Hello, our topic is on frogs and skin pigmentation changes. I'm Cassie.
I'm Mathea.
I'm Kayla.
And I'm Lily.
In our final review paper, we want to investigate the mechanisms of skin pigmentation changes in frogs, as well as understand the adaptive significance of those mechanisms. The paper we will be discussing in this presentation looks into pigmentation changes in frogs on a cellular and molecular basis. It looks deeper into the physiological mechanisms and the structures that contribute to how tree frogs change their skin color.
This paper looks specifically at the Rhacophorus dugradii, which is a type of tree frog that adjusts skin tone to its environment. It is an ideal model for studying vertebrate responses to background colors due to it having an excellent foundation for single-cell sequencing research. To answer the question, why do frogs change color, there are three reasonings for this. The first one is camouflage. This allows animals to blend into different backgrounds and avoid predators. An example of this is that a predator may not be able to see the frog if it blends into the leaf that it's on. The second reasoning is thermoregulation. Darker colors absorb more heat, whereas the lighter colors tend to have decreased heat absorption. Frogs are known to be ectothermic vertebrates. This means that they rely on external sources to regulate their body temperature. Background color matching is essential for camouflage and thermoregulation in these frogs. By carrying these out, they are able to increase survivability and therefore fitness. The third reasoning is communication. Animals and these frogs are able to communicate their coloration to attract mates. This could also increase the survivability and therefore fitness by their ability to reproduce and add on to the next generation. This is made possible through chromatophores, transcription, metabolism, and the immune system that we'll cover later on in this presentation. Within this paper, it focuses on two forms of color change, the first one being morphological color change. It involves the differentiation, proliferation, and development of melanophores, which can lead to changes in density and distribution of the pigments within the skin. This plays a large role in the production of melanin, which is the skin pigment, and the function of the melanophores, which is the pigment cells that contain melanin. This is thought of to be a more long-term or permanent adjustment. On the other hand, physiological color change is driven by the transport, dispersion, and aggregation of existing melanin within the chromatophores. This utilizes molecular components including cytoskeleton, motor proteins, cargo proteins, and the cargo recognition protein in melanophores. Under natural conditions, the physiological color change typically occur between minutes and even weeks on a more short-term time scale. And why do these matter? It's important, having two forms of color change in these frogs are important because morphological color change is a more long-term adjustment, whereas the physiological color change is both a rapid and reversible response. By having these two different forms, it allows the frogs to balance the short-term and long-term benefits. And by asking the question, do the status and activities of pigment cells differ among individuals exhibiting different skin color conditions, the researchers would like to comprehensively understand the factors driving color adaptation in relation to environmental cues. They are able to utilize single-cell RNA sequencing technologies to study this. The aim of the study is to investigate the cellular and molecular mechanisms underlying skin color changes in these tree frogs in response to the black and white backgrounds.
Before conducting the experiment, the tree frogs were acclimated to an artificial climate incubator for 7 days at 20 degrees Celsius with a 12-hour light and 12-hour dark cycle. The tree frogs were then randomly divided into black and white boxes, with the other conditions kept the same. The morphology and skin color of the frogs were recorded with digital cameras hours after acclimation. This figure shows the results, with the skin color of these tree frogs changing dramatically in response to environmental background color changes over 2 and 3 days. On the white background, the frogs showed a mostly green skin color with white spots under light conditions. In dark conditions, still on a white background, they were mostly green with brown spots. The frogs on the black background showed a deep green or black skin color, both in light and dark conditions.
Okay, let's dive into chromatophores. Chromatophores are pigment cells in ectothermic vertebrates that generate color by producing pigments or reflecting light. They facilitate dynamic color changes, allowing for adaptation to environmental conditions. The main driver of color change is chromatophore number. This article specifically focuses on the tree frog, Rhacophorus degradii, which has the ability to shift from light green to black skin based on their environment. There are two distinct chromatophore types, melanophores and xanthophores. They are responsible for the frog's coloration. Melanophores cause darker coloration and xanthophores cause a more yellow-orange coloration. You can see where they're located in the frog's skin of the figure on the right. The dominant mechanism behind color adaptation is the distribution and the amount of chromatophores. This article suggests that increasing or decreasing the amount of chromatophores plays a more significant role in color adaptation compared to changing pigment production in each cell.
Now looking at chromatophore numbers, according to results, specifically figure 5b from the paper, it was found that dark-colored frogs had higher proportions of melanophores and xanthophores in the skin compared to light frogs. Specifically, it was found that dark-colored frogs had 1.78% of melanophores and 1.25% of xanthophores, where light-colored frogs only had 0.1% melanophores and 0% xanthophores. You can see these percentages displayed in the figure on the right.
Both melanophores and xanthophores play critical roles in background adaptation in tree frogs, with their relative abundance corresponding to the observed skin tone. This suggests that an increase in chromatophore number is a key process contributing to the darker skin color observed under black background conditions. Melanophores from dark-colored individuals tended to exhibit higher transcription levels of tyrosine, TRP1, and pre-melanosome protein compared to those from light-colored individuals, though these differences were not statistically significant. Figure 6, shown to the right, shows how in dark-colored frogs the proportions of melanophores and xanthophores in the skin were significantly higher at the cellular level than in the lighter-colored frogs. These two chromatophores exhibited distinct gene expression patterns, especially those involved in their respective pigmentation synthesis. However, once again, this was not statistically significant. The figure shows transcriptional levels of core genes involved in pigmentation in melanophores.
Okay, let's move on to metabolism. Frogs are known to adjust their metabolism to match their color state. Distinct cellular functions are linked to different color states, light versus dark. Darker colored frogs appear to show an elevated energy metabolism in their melanophores, which indicates it takes more metabolic energy to maintain the dark color. Individuals exhibited higher transcriptional activity in energy metabolism pathways, and mature chromatophores have better potential for protein synthesis and energy production. In addition, frogs also adjust their immune systems to match their color state. Lighter colored frogs exhibit a higher expression of immune-related genes, since with immune regulation, it requires less pigment activity. It is theorized that this mechanism may arise because light-colored animals are more susceptible to UV damage. The researchers believe that these findings could indicate that amphibian color adaptation involves not only visible changes in skin pigmentation, but also underlying biological processes.
Now looking at limitations in the literature, only male frogs were sampled for this study to minimize the effects of sex differences on hormone levels and gene expression. This means conclusions may not be able to be generalized to female tree frogs. Also, only adult tree frogs were studied, which means these findings may not apply to younger frogs. The sample size being 30 also appears fairly small, and this reduces replications and weakens the reliability of the study. Lastly, only one species of tree frog was studied, which limits generalization of findings to other frog species and overall other amphibians.
Lastly, potential new research ideas. This paper advances our understanding of the mechanisms underlying color adaptation in amphibians, but also offers valuable insights into adaptive color changes in other non-modal vertebrates. Here we have 3 different areas of future research ideas listed. The first one, focus on the long-term roles of these mechanisms in facilitating animal adaptation to environmental changes. The second one is these findings could potentially hold significant implications for the conservation of endangered species. And the third one is biomimetic applications, which could expand our understanding and potential for leveraging biological adaptability in biomedical and technological innovations. Here's the citation for a research paper utilized within this presentation. Thank you for listening.